All right. Well, thanks everyone for being here. That's a tough act to follow. A round trip around the universe. I'm going to be a little bit more humble about my aspirations. So going to be talking about Full Tree 1 and 2, and it does dovetail into some of the topics that were just covered because I'm looking at evolutionary relationships through the lens of structure. We know that this approximation of looking at structure as just a chain of characters is inappropriate because the environment is actually what's interacting with these structures and putting pressure on them. We can't just look at them as a flat little sequence. They're actually 3D shapes. They actually do their function in this form. We heard a lot about that just a little while ago. So we need to incorporate this information into the way we do phylogenetics. So this is the problem that I'm trying to solve with Poultry 1 and 2. One of the motivating examples that kind of led me to explore structural phylogenetics and the evolutionary relationships between structures was this paper that was focused on gamete fusion proteins. So they fuse membranes, and they are present in most lower eukaryotes and plants, and in a lot of protostomes. Presumed to be in Leica, and also present in a lot of different viral lineages. We were able to discover another lineage in Archaea and actually get it crystallized. And funny enough, to help us phase the crystal data, John Jumper actually helped on this paper. This was pre-AlphaFold 2, but we knew they were able to sort of get structures reliably. We had trouble with the crystal, and he helped us out with this. We made some preliminary structural trees and got a lot of pushback from the reviewers. We were saying, like, there's no way we can align these in the sequence space. They have no sequence identity. Obviously, they're homologous. They come from a common ancestor. They're under the same evolutionary pressure to keep this function, to fuse membranes. They're all functionally equivalent. So we started making structural trees in this paper, and there's some preliminary work on that, but it's not really the focus of the paper. And just kind of responding to the criticism that we got in this paper and kind of going further with these ideas, we said, okay, well, they say there's no signal in this structure related to the phylogenetic relationships between these proteins. We're going to try and prove them wrong. So we made Fold Tree 1 based off of FoldSeq, which I'm sure needs no introduction to this audience. It's a very fast and efficient program to compare structures or search within huge databases of structures. And in this case, we just used it to compare sets of structures that were homologous in order to derive trees. And we made large data sets of AlphaFold structures within protein families that were homologous to benchmark this method. And in this case, we also included other methods which were sort of partially based off of FoldSeq, and as well as, the typical maximum likelihood approach. So this is available. You can try it on a Colab notebook. There is a paper out coming soon, Nature Structural and Molecular Biology. So keep your eyes out for that. So yeah, how do we actually benchmark trees? Because we don't know what evolution did during, we don't have a time machine. We can't go back. So we make an assumption. We say in a eukaryotic evolution, usually you just inherit vertically. You can't really do lateral transfer or incomplete lineage sorting or that type of stuff. In most gene families, you don't see this. Most of the eukaryotic mode of evolution is going to follow a logic kind of like in tree A. In tree B, we know, what we know about vertebrates kind of says that tree B is very unlikely, and tree A represents an evolutionary scenario that we can believe. And how do we do this? Well, we can label our trees. So if we have, for example, a set of leaves on the tree, each of these will have, a genome of origin, and we know the taxonomic lineage. So kingdom, phylum, class, order, stuff we learned in high school. And then we can say, okay, that lineage represents a set. And as we go up the tree, up to these internal nodes from the leaves, we can intersect these sets all the way up to the root and calculate a score which represent how well our tree agrees with the known taxonomy. And if our tree in eukaryotes agrees with the known taxonomy, we can say that's a good tree. And if this assumption holds over thousands of trees, on average, the method getting more signal should be able to make more coherent topologies. So if we do this over, in this case, 3,500 trees of homologs that are close enough to be identified with sequence, we can see that if we add some structural information, we're doing well. So if we explain this plot a little bit, the red squares are things that are distance-based trees. On the top, we have our evolutionary model. And on the bottom, we have the technique that we used for alignment. So fold-seq is using 3DI alphabet as well as amino acid. And our model here is just comparing the amino acids after fold-seq alignment. The state-of-the-art maximum likelihood model, here we see it in teal, those are all maximum likelihood models, uses amino acids in the evolutionary model and amino acids to align. So we used different structural distance metrics as well as different ways of aligning and different evolutionary models. If we move a little further away in the homology landscape, see proteins that are just structurally homologous in the CATH data set, where they're just classified by similar folds, similar architecture. The structural methods kind of gain on the amino acid-based methods. So we're adding signal, and when we see kind of the proportion of trees where different methods do better, you are definitely going to do better in this data set if you use structure. So we did a little case study on a protein family that was particularly difficult, just like the case study we did for the first paper with the fusion proteins. These are called the RRNPPA family of quorum sensing proteins, where we have an expert in the audience, Charles Bernard. He's the co-author on this paper. And actually, they discovered each of these families independently by crystallizing the family and realizing that it was the same as the others. There's basically no sequence homology between them. Very difficult to make a tree. And we see that different domain architectures emerge parsimoniously along this tree, and it has different aspects that are also kind of parsimonious and nice that are discussed in the paper. But yeah, that's not what I'm here to talk about. In this case, I want to talk about where do we go from here? Because we just have distance trees. And people were asking, well, we want to work with maximum likelihood. We want to be able to infer ancestral sequences, ancestral structures, have bootstrap support, have all these nice features. Yeah, it can't just be a brutal distance metric. We need to go a little further. So here's my one-slide introduction into sort of how I'm going to do this. I'm not going to go for information theoretic underpinnings. I'm an engineer, I'm not a mathematician. So I'm going to use machine learning and treat the structure as a graph. I'm going to use a little aggregation function around each of the nodes that I want in my graph to sort of create an embedding for. So I'm going to use the information of my neighbors and pass that through a learnable function. In that case, those are the little boxes there. So if I want to update sort of residue A in my structure, and these are my contacts in my structure, I'm going to take the neighborhood B, C, and D and update those and then update A. So this is a network with two graph convolutions. So it's going for the neighbors of the neighbors. So in this way, we can train new discrete alphabets. So I'm going to create a graph from a protein, look at the contacts between the different residues. Anything less than 10 angstroms, for example, I can consider as a contact and an edge in my graph. And then I can label each of these nodes with lots of features. So in this case, all the physical chemical features, the bond angles, solvent accessibility, whatever you feel like. So yeah, this is also part of the project, selecting informative features. And I'm not going to go into the architecture of the network that I used, but I have an encoder network, which will basically turn each of these nodes into a discrete state of an alphabet size, which I specify. And then from there, I would like to recover the amino acid identity of each residue, as well as the contact map between the residues. And if I can do this well, I'll be learning something about protein structure and sequence and storing it in this alphabet. So here is a 100 epoch training run of a very small network. So it's about less than a million parameters. Just to give you an order of magnitude, AlphaFold is 21 million parameters. And it's learning to encode these discrete states. And then this is one particular example protein where it's encoding the protein after every epoch. So it's trying to make this protein in this alphabet, in this sequence of characters. And then it's going to use a local window along that protein and try to determine which residues are in contact with which, and what is the identity of the amino acid at each position. And after a while, it sort of converges to a logic where it decides, okay, I'm going to give each of these positions a specific identity in my new alphabet, which allows you to learn a little bit about the structure and the sequence as well. So it's got that information stored in there. On that second row, we see, on the first row, we see the contact prediction of the network, as well as the original fold distance matrix and the contact points, but they're very close to the diagonal in this case. Can't really see them well because of the compression on the video. And then we have the ROC, precision recall, and the distance, well, the normalized distance between the residues, as well as the probability of contact. So there's some correlation there, but it's training for contact and not distance. And then we also have the ROC off diagonal, so 10 off diagonal, as well as the prediction of the different characters that it's going to use. So here we're going to break some assumptions that we're making about structure and go back to the sequence sort of paradigm, because we're going to try to make something retrocompatible with existing tools. And I would like to make a substitution matrix to be able to align these new sequences and then get trees for them. So we have about 3,500 protein families from the AlphaFold cluster DB, where we're just aligning using fold-seq, because we believe those alignments, they're using structural information. And then we're going to make a substitution matrix by reading off the number of sort of swaps that we see over this data set. And now we get to measure how well it does. So our buddy John says, make sure you study something where you have measurable ground truth, and something that's really important and that's got an impact. But unfortunately, I study, phylogenetics, so that's kind of a tough sell. But we have the TCS, so we're going to run TCS on it. Fortunately, it does a little bit better on the CAF data set. So we've got a slight boost on the CAF data set, and we're doing slightly worse on sequences that are closer. But this is just a prototype where, as John was talking about, taking out modules, putting modules back in, deciding which sort of alphabet size I'm using. It's definitely a work in progress, but yeah, it's getting there. It's giving good trees with good topology. And since we'd like more benchmarks and more measurable ground truths, I'm proposing a few new approaches now that we have maximum likelihood. So we're going to infer some ancestral character states and then try to go back to the amino acid and fold those and see if they do better than the amino acid-based reconstructions for those particular internal nodes in deep phylogenies. So we want to see when the folding logic breaks down. We know that AlphaFold has only been trained on foldable structures, things that are out there in nature. So it's just kind of a rough guesstimate of whether or not these things will actually fold or how uncertain the model is. But it'll give us a grasp of whether or not our model is doing a good job with structural sort of coherent, structurally coherent sequences. And then the second one, we're going to make a big species tree with a concatenated alignment and go over each column likelihood and see whether or not the proportion of character states in each column is informative for our tree likelihood. So we're looking at the mutual information between our character set and our tree likelihood for a tree that we believe in this case, the species tree. So yeah, there is a preliminary pipeline under heavy development available, which will take you from a set of structures to an encoded sequence, allow you to align them with a substitution matrix, and make a tree from that. For the particularly brave and not faint of heart, you can also try training your custom models with different architectures and different character sets or different input features. And I would like to be able to, from these character sets, infer folding energies. So that's almost done. So in the FoldX style, as well as be able to output directly a proposal for a 3D model of the fold tree 2 sequence. So have kind of a folding module like an ESMFold or AlphaFold, where you just kind of get a 3D model. Probably going to be very coarse and not doing the best job, but it's something I would like to try. And also, since we know that it can get contacts within a sequence, it would be nice to try between two sequences. So complexes or dimers or things of that nature. So yeah, I'd just like to thank everybody for listening, and if you have any questions. And yeah, also some of my collaborators, Pablo Aguilar, you might know him, collaborate with a few people in Argentina. So yes, definitely pay attention to the science in Argentina and support them in any way you can. There are a lot of talented people out there, and yeah, it's worth putting that forward.
We have time for questions, actually quite a few minutes. Questions. I have a question. So I read this paper, I think it's great work. I read one separate print. I think it's a great idea, but it assumes that you are working always with homologous proteins. I mean, what the caveats are if, I mean, it assumes that basically there's little convergent evolution, right? Because you're taking your inputs based on the positive alignments. So what's your, I mean, are you applying some heuristics or something to check for that?
So when we're looking for convergent evolution, like I've seen a lot of papers where they're trying to prove that two sequences will end up in the same fold from different ends of the sort of sequence space and converge in the structure space. You typically see that in just a few edge cases, a few pathological edge cases, like coil-to-coil domains, or things that are very low information, let's say. Arriving to that just randomly is something that people have yet to prove. Once you get to a complex enough architecture and you're looking at an alignment, structural alignment over hundreds of residues, for example, what I showed in the beginning of the presentation, the fusion proteins, arriving at that by pure chance when there are several different fusion protein families which are functional is very unlikely. Why would they converge to this? There are lots of ways to skin a cat, let's say. So that's just a sort of qualitative way of saying it. But other than that, the protein families I was working with in this benchmark are things that are kind of known to be alignable over a certain percentage of residues of the protein and have the same architecture. So there's no junk going into the benchmarking. Yeah.
Any further question?
Thank you very much. Really interesting project. And sorry if my question may be a little late, but if you have a protein structure, you have the taxon of the organism of this protein structure. Yeah. And from the taxon, you also have the phylogenetic tree. So is it a difference between the tree of the organism and the tree of the protein? And does it make sense to distinguish the two or give some advantage?
The taxonomy, the taxonomic tree will often tell a different story than the phylogeny of a particular family. The taxonomy can kind of be thought of as the compilation of all of the phylogenies. Like I was showing in the earlier slides, there is a possibility even during vertical descent to have duplications and losses, even in very simple eukaryotic evolution. If you're going towards prokaryotic or viral evolution, you have lots of lateral transfer, you have domain shuffling, you have all sorts of phenomena that don't respect the species tree at all. So just because you know where that particular protein came from doesn't mean that there's no interest in contrasting the phylogeny with the taxonomy, especially in cases where the evolutionary scenario can get complicated, like in the first example that I showed. Viral and eukaryotic lineages kind of spinning off of archaea, where in archaea it's actually in a mobile element and not respecting the species tree or core genome at all. So to know that, you would need to do the phylogeny of these things, and you couldn't without using structure.
While we have next question, can the next speaker please go and start to put the slides so we don't get so...
Great talk and great work. Similar to the previous questions, so with viral glycoproteins and all these kind of things, you get extensions quite often. So then you have a core structure and then you have something kind of sticking out on top.
Mm-hmm.
Have you looked at all into how that kind of extension looks in terms of convergence versus divergence with your with your phylogenetic inference model?
So you mean sort of domains getting added?
Yeah, so if one thing follows a specific evolutionary pattern in the structure or the core, and then two things have convergently added a bit of the top. Yeah,
we did actually look at that for the RRNPPA family. That was part of what made us believe this tree was actually believable. So there are different domains. There's a core domain, and then there's TPR domains that are added on. And, there's different numbers of these repeats. And then we inferred kind of clusters of these different domains. So we chopped the things that were not sort of conserved across the entire family off. And we did the phylogeny shown in the slide on the core domain. And then we said, okay, where do these extra domains appear on the phylogeny? And it looked like they appeared just once, which is very parsimonious. You say, okay, there's not like multiple times where they show up or whatever. It could be, I mean, that's not a scenario with zero probability. But in the one that we showed, we see that they tend to emerge once and be conserved as they're advantageous for the organism. So that's typically. Oh, sorry.
Time's up, please.
Yeah.
Move on. Thank you.